The broadcast is now starting. All attendees are in listen-only mode.
Okay, good morning, everyone. We'd like to welcome you today and thank you for attending our webinar on tissue process validation concepts. The intent of our webinar is to provide those who perform or review process validations with the concepts and knowledge needed to properly set up the validation and interpret the data. The webinar includes concepts which are also addressed in the upcoming Microbiological Surveillance document from AATB. And here to present are Martel Winters and Alpa Patel, our resident experts here at Nelson Labs. Just as a little bit in the future, for some news on Nelson Labs, you can always refer back to our webinars page if you miss it or would like to refer back to it. We try to have our webinars up about a week after the event, and those are under the Knowledge Center on the www.nelsonlabs.com website. You can receive all future email notifications about future events and webinars by following Nelson Labs on Facebook and Twitter. As for the webinar, we welcome all of your questions, and you can submit them anytime through your questions tab. Martel and Alpha will randomly answer as many possible questions during the last 15 minutes of our event today. Now I'd just like to welcome and tell you a little bit about Martel and Alpha. Martel Winters received a Bachelor's of Science in Microbiology with a minor in Chemistry from Brigham Young University. He has been with Nelson Laboratories for 17 years. He's currently a senior scientist for Nelson Labs. Martel serves on the AAMI Microbiological Methods, Sterility Assurance Level, Radiation Sterilization, General Criteria for Sterilization, and Terminology Working Groups. He is also currently heading the AAMI Task Force for a document regarding sterilization of tissue products. He is on the AAMI Sterilization Standard Committee and on the AATB Standard Committee. Alpha specializes and consults with many clients about cleaning, disinfection, and sterilization on reusable medical devices. Her role as department scientist, she is responsible for writing validations and tissue studies, designing and operating in-house validations and R&D testing, assisting with company-wide training modules, and mentoring and co-leading the Nelson Labs Sterilization Specialist class. Alpha is a member and collaborates with the ASTM, AATB, and AAMI standard committees. We would like to welcome Martel and Alpha, and the time is now yours.
Thank you, Mike. This is Martel. I'll be starting. I'm going to go over some of the initial concepts regarding the tissue process validation. I'll go do a quick review of what the standards say about validation, talk about some of the concepts from the Microbiological Surveillance document that Mike mentioned, and also discuss some details on process characterization and process validation, and then discuss some process validation approaches, and then Alpha will go into more detail on the process validation approaches. So there are really two main areas that we look to for requirements regarding process validation. The first, of course, is the FDA and their 21 CFR Part 1271 Subpart D. And process validation, in general, it says, where the results of processing cannot be fully verified by subsequent inspection and tests, you must validate and approve the process according to established procedures. Now, you see the requirement here is fairly loose. There is the requirement to do it, but no requirement regarding how to do it. So the established procedures are determined by the individual tissue bank. And all you have to do is just show that you are accomplishing what you want to accomplish. And then clause B on written representation from that same area says that any written representation that your processing methods reduce the risk of transmission of communicable disease by an HCTP including but not limited to a representation of sterility or pathogen inactivation of an HTTP, must be based on a fully verified or validated process. So again, it's just saying that whatever you say you do, you have to demonstrate that you truly can do it. And some of the key words here, you have a fully verified or validated process. And what we're trying to lean toward here is a validated process rather than a fully verified. The way I interpret that is a full verification implies you're doing a lot more testing of finished tissue than we would prefer in most cases. So a validated process allows for greater process monitoring rather than tissue testing. And that's something that I frequently am trying to preach and make sure is clear in the industry regarding that option. Another thing that we rely on heavily, and I would strongly recommend that you review this if you have not already, is this is FDA's document of guidance for industry on process validation on general principles and practices. And this is fairly new, just in January of this year, and it's an excellent document that discusses the various some of the main concepts in process validation. It's a very helpful document. If you're not familiar with it, then you should. From an AATB standpoint, the 12th edition currently says it talks very little about process validation, and mainly just says it mentions a need for QA to ensure that process validation studies are performed as necessary and that they're properly reviewed. So it's very open, and AATB really should not have to say much more than that. You know, that the requirements for process validation are already mentioned in FDA and the FDA standards that I referred to, and so additional requirement really is not necessary. Additional guidance may be helpful, and currently there's no real guidance or requirements from AATB, but that's something that's been in the works for perhaps close to 3 years now, and hopefully the microbiological surveillance document will be able to be out fairly soon, and that can provide some additional guidance and some examples on how process validation may be accomplished. So this guidance document from AATB discusses the importance of process characterization as an early part of process validation. And that's something that is discussed pretty well in the FDA guidance document that I referred you to. And I think this is something that is largely overlooked in the tissue industry, where sometimes the process characterization is not addressed as really as it could or as it should be before people go right into process validation. And it also, the guidance document also provides some concepts and examples of proper process validation or thorough process validation, as well as some guidance regarding test method validation and qualification. And there are a lot of other details in there as well, but this is what we have. This is what the main points that relate to what we're talking about today. This is something I mentioned briefly, but I want to mention again that one main point, and I was very involved in writing this document. One of my main goals was to try and provide whatever information we could, so that as we are developing and performing process validation, that the focus can be more on routine process monitoring rather than routine product testing or routine tissue testing. And by routine process monitoring, those are usually the items that you would monitor routinely from the process. Those would be things that you could that you would determine during process validation. Benefit with thorough process validation that you would be able to determine which items or which steps of the process carry a larger or carry more weight in the finished result of the tissue from a microbiological standpoint. And that would, if you can determine which those main points are in process validation, and those will be the things you monitor in processing, rather than ending up doing a lot of tissue testing at the end. So in process characterization and validation, we'll talk about this a little bit. And this is a flow chart that I've put together, and you can't see all of it very well here, but I'm going to zoom in on parts of it here in a minute. But this is just to help describe to some extent what kind of things to consider in process design and process characterization before we step into full validation of the entire process together. So just to zoom in on the upper portion of that, since this is more the part that I think that some areas in the industry could, we can focus on a little better, is in the process design and the process characterization aspects of validation. And I'm not going to go through every square here, every step, but just to help understand some of the concepts here, that I know that it is nice and it's easier in setting up a new process or bringing a process into a new facility or a new clean room, that it's very common to want to do what you're familiar with or what you've done at previous companies or what seems to make sense. And there's nothing against taking that approach, but if we address a few things prior to full implementation or full validation, then that can really simplify or that can really improve the process itself as well as simplify the final validation. So these initial steps, we're talking about understanding what each step is supposed to do, and if there's supposed to be a microbiological component or microbiological aspect to that step, that it should be considered, or in most cases at least some degree of testing going into that to determine that it is performing as it's supposed to. And in this situation, I'm not trying to address every aspect of process validation or of a process, a tissue process. I'm just talking about the microbiological aspects. So if each step is broken down into what microbiological benefit you are supposed to get from that step, then those individual aspects can be considered, and if it is an important part of the process, as far as if that step is supposed to provide a particular log reduction or be beneficial against a specific type, a specific set of microorganisms, then it's beneficial and wise to make sure that you are that you know ahead of time, based on just that step alone, what type of benefit you're getting. So the top row of this flow chart is meant to just provide some thoughts regarding that aspect of process design and process characterization. And you see in the upper right-hand corner there which steps or components need to be fully validated, where the note there is that qualification testing, qualification versus validation, qualification tends to be something less stringent. For example, you might, like I say there, you might only do it on a single batch versus 3 batches or 3 runs. But also qualification might involve a more simple test scheme rather than a full validation. And not all components or not all steps really need to be fully validated. the main point I'm bringing up here is you just want to understand what each step is doing and make sure that each step is functioning properly before they're all put together and before the process is evaluated as a whole. The second row down, going from right to left, talks about, you see the second box over, should multiple variables be evaluated? So in this case, oftentimes the microbiological benefit from a step may hinge on multiple variables, not just one. For example, it might be the amount of time that the tissue soaks in that solution. There might be multiple concentrations to consider. Maybe different types of tissue may react or may get different results from that soaking step or whatever that step may be. Temperature, pH, there may be other aspects or other variables of that step that need to be considered. Now, usually I try to recommend that you not in evaluating a particular step, I usually try to tell people not to evaluate different times and different temperatures and different pHs and different concentrations, but a couple of those that either from experience or from information from the supplier where you feel that you might have a pretty good handle already on what that particular variable should be set at, then just set that. And then the ones that perhaps you're not as comfortable about, for example, time or temperature or concentration, then you might use different time points or different concentrations, for example, as part of your initial qualification work or your process characterization to help you determine or understand if playing with those provides better results under certain conditions. And once you determine what those variables are that you want to evaluate, then do perform evaluations on those. And you see there's kind of a loop there. It's kind of it can be kind of an iterative situation where you may try a couple of conditions with one aspect or multiple aspects and find that one of them works well, but others might need to be modified to provide better results. And so you may need to go back and perform some additional evaluations of that as you optimize that particular step. But you see the final box down there on the bottom, based on data and testing, should the step or that component be optimized? And if no, then you'll go down. I'm going to go up one slide here. Then you'll go down into, okay, now you fully understand each step of the process, and you understand the variables of each step, and you've optimized these steps to provide the best benefit that you need from that step, and now put it all together and perform an evaluation on the entire process and see if that meets your criteria. Okay. Now, oftentimes when we're talking about process validations, a very common, from a microbiological standpoint, a very common term is a log reduction. I want to make sure that concept is clear before I go into my next slides and before Alpa goes into hers. So the term log reduction just means how many microorganisms are being either killed or removed by the process. For example, a 3-log reduction means that you have killed or removed 1,000 CFU. And one thing I do not have on the slide here but want to point out is that I need to make it very clear that a 3-log reduction does not equal a 10 to the minus 3 sterility assurance level. The concepts are related, but the numbers are not the same. The log reduction tells you what the process is capable of, and the sterility assurance level, if that is something that you're referring to in your process, that is based on both the log reduction capability of the process and based on the initial bioburden on your tissue. But the log reduction or the process log reduction is just what helps you understand the microbiological effectiveness of the process. And that is usually focused just on microbiology. You usually do not use the term log reduction for any other aspect. So now I wanted to go through some of these scenarios that are common, either are or have been common in the tissue industry regarding process validation. And one of the first ones is the example where oftentimes you may just take tissue, maybe just typical tissue, or maybe something you might feel is kind of worst case tissue, and you put it through the proposed process and test for sterility afterwards. Now that is very simple. It seems intuitive, and it is undoubtedly it is very easy, but really in most cases you can gather very little, or in most cases actually almost no real understanding of the capability of your process when taking this approach. The reasons for that are you have to ask the question, so to really understand what your process is capable of, so what was the microbial count at the beginning on the tissue? And in most cases you don't know what that was if you're just taking normal tissue and putting it through the process. And then what was the microbial count at the end? Well, if it was sterile, if it came up with no growth, then you know that there were 0 CFU. That's a safe assumption. But any that have growth on them, you have no idea whether that was growth due to one remaining microorganism, or it could be 1,000 or 10,000. You have no idea what that final count, microbial count, is. And without a starting count and without a final count, you cannot determine the number of log reductions. And that means that you really have no idea what the microbiological capability of your process is. And also, when you take this approach where you put tissue through the entire process and test for sterility, you also don't understand what each step is providing. I'm assuming in this scenario I'm talking about here that thorough process characterization has not occurred prior, because usually when this approach is taken, it is because there's a lack of understanding of what a thorough process validation should be like. So in this scenario, you also don't understand there may be a situation where one step in the process is providing all of the microbiological reduction, and the other steps are not doing anything. Now, the next step up would be a situation where you are inoculating the tissue with specific microorganisms. So in this situation, you know what the initial count is, and you know the microorganisms that are on there, and you put it through the process and then test for sterility. Now, let's say you have, you know what you started with, for example, 10 to the 5th CFU, and what is the final count? If you don't see any growth on those sterility test samples, then it's safe to assume that there were 0 CFU. But again, any growth that you see, you have no idea whether that was 1 or whatever quantity that may be. So again, in most cases, it is still difficult to determine a good log reduction. If all the samples come up sterile, or if only a couple of them grow, then you can calculate a log reduction. But you're hoping that you get all no growth to take this approach in order to get information you can really work with. So the ideal situation, and I'll just go into it briefly, and Alpa will go into it in more detail, would be where you start with process characterization, which I've discussed. That way you really understand each step of the process, and you actually have data with most of those steps. And then you've had a chance to optimize each step, so you have a really good understanding of what the full, not only just what the steps should provide, but what the full process can provide. And then with an understanding of the CFU at the start and at the end, where we're talking about performing a bioburden test rather than sterility testing at the end, and Alpa will discuss that in more detail, then you get really good information regarding log reduction capability of that overall process. And like I said, Alpa will provide an example of this. So that is the end of what I was going to address. And like Mike mentioned, we'll take questions at the end. So I will hand the remainder of the time over to Alpa.
Thank you, Martel. For the second part of the webinar, we will focus on the specifications of tissue testing and the steps that are taken to perform this validation. In this part of the presentation, I will go over the process validation testing and how it is performed through the eyes of the laboratory. I will go over specific testing details of the bioburn and sterility testing and the differences in the testing composition and the steps needed to accomplish the validation, including sample size and type. To go over some points that Martel specified, process validation is a term used to validate step-by-step methods and procedures that are used to process products like tissue. Most of the process validations are performed in-house by the tissue banks, but if a facility is interested in testing their procedures through a third-party testing lab, they would have to provide all the steps that are performed for the processing. By contracting a lab to perform the testing, it not only gives you a microbiological process, but gives you how well the tissue process is performed by testing every step of the process. Here's an example of a test flow that are commonly used during tissue process validations. All the steps in the test flow are performed for the processing, and then the tissue is tested for sterility at the end of the process. By doing process validations, you can break out the steps and then test them individually or in a group and find out if all the steps are necessary for the processing and at what point the process does the tissue become sterile or free of organisms. For example, if there are several steps with antibiotic or disinfectant rinses, it is better to know first how much microbial reduction you obtain in each step. This not only helps you to optimize or eliminate the process steps that are not necessary, but decreases your processing time and making the processing more efficient, in which Martel has gone in detail for the optimizing or eliminating the process steps. Before we can execute a validation, it is important to have a protocol in place. This protocol is usually written by the laboratory, but the step-by-step process for the tissue processing has to be provided by the facility, and the method they wish to evaluate the process also needs to be decided, whether it be through bioburn or sterility testing. The selection of organisms to be tested also needs to be established by the facility, and this information may come from their environmental screening or recovery methods. For the test flow for bioburn test approach, the first and the most important step is the neutralization step. You have to make sure that you perform the neutralization before you do any of the testing. The reason for performing the neutralization is to make sure that the chemical or the antibiotic cocktail used for processing the tissue samples is neutralized appropriately. If you don't perform neutralization, false negatives can be seen. USP recommends to achieve a 70% recovery compared to the control. Once the neutralization step is accomplished, then process validation begins with the contamination of the samples with the selected organisms, and then putting the samples to the chemical or the antibiotic soak. Once they've gone through the processing, the extraction performs with the samples in the neutralizer and follows with the bioburn analysis using selective and differential agar plates, and then these plates are incubated at a specified temperature for the organism, tested and then enumerated. This process gives you a log reduction value by comparing the positive control titer with the processed tissue count. As for sterility testing, it is almost the same as the bioburn analysis. The only difference between them is the samples do not go through the extraction phase. The samples are sterility tested right after the chemical or the antibiotic process in a sterile container and then incubated for 7 days at a specific temperature for the organisms that was tested and scored for growth at the end of the incubation. This process gives you a pass or fail or a growth versus no growth result. Once the devices are contaminated, the samples goes through an antibiotic or a chemical soak, which is the most common method used in the reprocessing for tissue validations here. As you can see, here's a list of antibiotic chemicals that have been used for the process validations. Most of the time, an antibiotic cocktail is used for process instead of individual antibiotics. It seems by mixing the antibiotics together, it seems to give a better microbiological process. Although this is one of the most common methods used for reducing contaminants, it usually does not tend to kill organisms to the level expected for the process. It usually gives 1 to 3 log reduction and is not very effective. It seems that chemical disinfectant tends to work better in killing or reducing the amount of organisms on the tissue samples. We have seen from a log reduction that varies from 3 to 6 log reductions, and it seems like chemicals are starting to take over the antibiotic cocktail disinfection methods, and usually are used for sterility testing as well to have your products sterile at the end of the process. When you're considering picking organisms for your validation, it is wise to test for organisms that are common environmental contaminants and show up in routine occurrences when screening the tissue. When in doubt, it is recommended to choose one organism from each of the following groups listed on the slide: a gram-positive rod, a gram-negative rod, a gram-positive cocci, yeast, mold, and a rope. These are some of the most common organisms that are tested for tissue process validations. Only one from each group are usually tested. If you want, you can test them all, but as you know, as you test more, the cost increases, so a lot of people usually choose one organism from each group to validate. The organisms can be tested individually or as a mixture. The results are more valuable if they're tested individually because you can get a log reduction value for each organism, and therefore allows you to know how well each organism works with the decontamination process. One organism that we recommend to test for is Clostridium, since this is a reoccurring organism that is a problem in the tissue industry right now. The number of samples used for the validation is critical, but there are no requirements right now for how many samples you should be using for the validation. Tissue samples submitted for testing has to be submitted sterile, which is usually performed through radiation. This is because if we test a sample that already has background contamination, it can obscure the final results and give false data. Validations are usually performed with 4 tissue samples per organism, in which 1 represents a positive control, which does not go through the process, and 3 represent the process samples that go through the entire chemical or antibiotic treatment. There are a lot of different size samples used for the validations, but there is no particular size distinguished out there yet that is required for the testing. But because inoculating is very difficult for small samples, we require at least a sample size of 1 centimeter by 1 centimeter. As I mentioned before, neutralization is one of the most important processes in the validation. If the chemicals are not neutralized efficiently, you can see false positives, and therefore you get false negatives, and therefore you can't rely on your results. Therefore, we always recommend to do neutralization for all our validations. This is an example of how we perform inoculation for our tissue samples. Most of the inoculations are performed using a sterile syringe in a HEPA-filtered hood. It is ensured that contaminants are not introduced during this process. There are several methods used for inoculating, but the most common method is used through direct inoculation. Direct inoculation is usually the most used method, but sometimes due to sample constraints, full immersion is the only way to go. How a sample is inoculated usually depends on the type and size of tissue being tested. Direct inoculation is used for tissue samples that can hold inoculum on the surface and are directly inoculated with small droplets of the prepared serum using a sterile syringe or a micropipette. And with this method, you actually know how much you have put on the device, and therefore it's easy to enumerate it. Full immersion, in the other case, is used when tissue samples can't hold inoculum on the surface due to small size, irregular shape, or surface tension, which we do see with a lot of tissue samples as well. Each sample is inoculated with a target inoculum of at least 10 to the 6 CFU of the organism to be tested. This titer is chosen to challenge the process in the worst case scenario. There are two test methods that can be utilized for the process validation. One of the methods is bioburn, which gives you a count and a quantitative analysis, and the other one is a sterility method, which gives you a qualitative analysis and gives you a result of a growth or no growth. The most common test is the bioburn method for tissue process validation. When you want to consider which method to use for the validation, you would want to consider bioburn analysis when evaluating individual steps, as Martel went in detail. This provides a log reduction value and helps you understand the effectiveness of the process or the process steps. It also provides recovery efficiency, which is critical to understand the effectiveness of the test method. When to use sterility is when data is available that the process provides a 6 to 7 log reduction, and initial data is usually determined using a bioburn approach in the first place anyway. If a 6 to 7 log reduction is not obtained, all samples will be positive for growth and a log reduction cannot be calculated. To go through the step-by-step process of a bioburn method, here's the step-by-step process for performing the bioburn method. The first step includes product inoculation, usually on 4 samples, followed by a dwell time, which is usually an hour. The reason for the 1 hour is because a lot of organisms tend to die off after the 1 hour period and therefore compromises the log reduction. Once the dwell time is completed, the positive control is extracted through an exhaustive extraction, usually 4 extractions on the same sample, and plated on appropriate agar through sealed dilutions and membrane filtration methods. Once the plates are plated, they're incubated and enumerated. The rest of the samples are processed with a chemical or the antibiotic treatment and then extracted and plated, same as the positive control sample. Once the colonies are counted from the plates, a log reduction value is calculated. Here is an example of how log reduction is calculated. You first take the log of the positive control titer. Now this titer is from the positive control that was not processed with the antibiotic or the chemical. So you subtract that from the log reduction on the recovered counts from the processed samples, and that's how you get your log reduction. There is no acceptance criteria right now in the industry that tells you at what level of recovery or log reduction is acceptable. This needs to be defined by the facility with justification of the acceptance criteria. The device industry has an acceptance criteria laid out for disinfection of reusable medical devices. Therefore, a lot of facilities that perform tissue process validation usually follow the device industry acceptance criteria, which specifies to achieve a 6 log reduction after the samples have been processed. This is a very common approach that we see with the industry with log reduction values. As for sterility testing, once the tissue sample is contaminated, they are processed through the chemical or antibiotic treatment and then aseptically transferred into neutralizer and incubated at specified temperature for 7 days. Once the samples have completed the incubation, the samples are scored visually and given a result of a growth versus no growth. If you wish for a log reduction value, MPN, which stands for most probable number method, can be used to evaluate the effectiveness of the process. But for this process, positive control must be inoculated and extracted to enumerate the initial titer of the sample through the bioburn method. This method is used for samples like powder or putty or paste of the tissue samples that we see and samples that cannot be filtered. Here are the steps of how the MPN method is calculated. You first take the natural log of the samples tested, divide that by the number of negative samples. If all samples are negative for the growth, assume one positive for the calculation. The log reduction is then calculated by taking the log of the initial titer of the positive control that you obtained by the Barber method and subtract that by the process samples, which you get from the previous calculation. This is a picture of samples that have been processed with sterility testing and have been incubated for 7 days, and you can see visually if you have a positive growth. It's very easy to distinguish between negative and positive growth. So this is an example of that. In conclusion, I would like to emphasize that process validations are very important and should be performed routinely. Process validations help to assure that the quality objectives and requirements of the product being used are achieved and provide confidence in the tissue process and assurance of the quality product. Thank you.
All right, we'd like to thank Alpa and Martel for their knowledge and their expertise today and sharing this tissue process validation concepts with us. We'd like to open up the time now for question and answer. You should be able to see a questions area on your side on your computer. Go ahead and just send us in your questions, and we'll get to as many of those as we possibly can. And I'd like to turn the time back over to Alpa and Martel.
Okay, we are organizing ourselves here so we can see the questions. Okay. I got a question about talking about how to select the challenge organisms. Alpha, you want to go through that, or you want me? Okay. Well, so in selection of the microorganisms, Alpha showed a nice slide with typical microorganisms that we see people doing a lot of. Now, there are not any requirements anywhere by FDA or AATB that particular microorganisms that specific microorganisms be addressed. But it is very common, and FDA is accustomed to seeing this approach where, like Alpa said, a microorganism is selected from each main microorganism type. For example, a gram-positive cocci, a gram-negative rod, a gram-positive rod, a mold, and a yeast. Now, it would be wise if you tend to see a specific gram-positive cocci frequently on your tissue, it would be wise to use that microorganism rather than some other one that you might just pick. So it certainly makes sense to, in selecting the microorganisms, to have a good understanding of what you tend to see on the tissue, either on your recovery cultures or what you tend to see coming through on any finished tissue testing you may have. And but if you want to evaluate, like she said, if you have a gram-positive cocci, if you want to address, for example, a skin microorganism like Staph aureus, but if you also see another gram-positive cocci and you want to address that one as well, you're certainly able to do that. But at this time, there is no expectation that the panel of microorganisms be selected in any particular fashion, or there is no requirement that it contain, or that list contains specific microorganisms. At this point, you are to make a rational selection based on covering all the organism types and based on what you tend to see on your tissue. Anything to add to that, Alpa? Okay. All right. Let's see. Another question you picked out there. I'm just reading through questions here. Please give us a moment. Okay, can we define BF testing and how this fits into sterility and bioburden? So the BF test is it's bacteriostasis fungistasis test, and that is what we use to demonstrate neutralization for the sterility test. So Alpa talked about the importance of neutralization, and the BF test is how we demonstrate neutralization for a test of sterility. And simply what we do in that situation is we take the tissue in the same form, so it has gone through the same process as what you will be putting the tissue through for testing, and you place it in a growth medium, in the growth medium that you think you're going to use, and then you add low numbers of certain microorganisms to that broth with the tissue in it to see if you can get growth. And if the low numbers of those specific microorganisms can grow, then that shows that you've properly neutralized the samples. Now, USP has a specific list of microorganisms to use for a BF test, but in this case, where you are inoculating the tissue with specific microorganisms, it is wise to use those in low numbers, which is usually less than 100 CFU. It's usually wise to use those to demonstrate neutralization in your BF, so that you're sure that each microorganism you've inoculated on and onto the tissue, that you can recover those if they do survive in low numbers through that tissue process. Alpa, anything to add to that?
No.
Okay. Okay. Question here about should mycobacteria or environmental or pathogenic microorganisms be considered in the microbiological panel? So another question on the panel that you select. There is no requirement that mycobacteria be used, and if there is potential for mycobacteria to end up on tissue that may come into your process, then certainly you would want to address that. But at this point it is not required, nor is it required that pathogenic microorganisms be considered. Often we like to use pathogenic microorganisms because those are the ones we're concerned about, but there is no requirement that every microorganism in the panel be pathogenic. The purpose for the panel is to represent a broad variety of microorganisms, and as you select the different types, if there happens to be one which you are concerned about from a pathogenicity standpoint, then you would be perfectly fine to select that one. But at this point, the pathogenicity issue is not a requirement in your selection process. It would be wise to do so. Okay, you're pointing to another one. Can you just show log reduction of spores instead of the panel of various microorganisms? Generally speaking, it is a safe assumption that spores are a worst-case scenario when it comes to disinfection or sterilization. So that if I could foresee a situation where you would have a good rationale in place to show that the spores would be a worst-case scenario and therefore use spores. The problem with spores is that with certain types of tissue, the chemicals will result in tissue that is not functional or not functioning the way that it should. And so for many types of tissue, those harsher chemicals are not an option. And selection of spores in that scenario will provide you very disappointing results and may be too much of a worst-case scenario. It is not extremely common to have high numbers of spore-forming microorganisms coming in on your tissue, and so that's where we discussed a situation where do we want to talk about a worst-case scenario, W-O-R-S-E, versus a worst-case, W-O-R-S-T. A worst-case scenario in some cases may be overkill or too much overkill and not necessary. So I can see where it would be an option. if you feel that the process would be capable of doing so, but I would not always default to always using spores because of that potential problem if your process is largely based on antibiotics rather than chemicals. Anything to add, Alpha? Okay. Any other ones that jump out? There's a question here, why is 10 to the 6 considered a worst case scenario when choosing bacterial load for the target inoculum? That's a good question. 10 to the 6 is obviously an extreme worst case regarding what you would expect to have the tissue actually have on the tissue that comes into your process. But in this case, we are not inoculating to represent what your tissue might have on it. We're using a high number so that we can understand the full extent of the capability of the process. If you only inoculate with 10 to the 3rd and end up with all kill with no growth on any of the testing at the end, then you know there's got a 3-log reduction, but what you might not know is you might be getting a 6-log reduction. So you don't fully understand the capability of the process if you inoculate with something lower than 10 to the 6. So we always recommend 10 to the 6 or 10 to the 7 just to give you a better understanding of that process. Okay, still going through here. Have you picked out any other ones? I'll just keep picking unless you point out other ones. Here's an interesting one. How do you approach process validation for aseptic processing when no claims are made for elimination of microorganisms? And sterilization methods are not indicated as cell viability is a key requirement of the tissue. So this is assuming where sterilization is not an option, but in an aseptic process, so that's a good point that's being raised there. In an aseptic process, you are not necessarily demonstrating, you don't have to necessarily kill microorganisms. The traditional aseptic process means that you are already dealing with clean materials and you are keeping them clean throughout the process. So for aseptic process validation, using the term aseptic process as FDA would see it, then you would want to refer to the ISO 13408 series, specifically Part 1 of that series, so 13408, Part 1 and Part 7, which is actually not available yet, but I'm part of the working group who's writing that document, and it provides good guidance on performing aseptic process validation when you don't have very high numbers of products to test. So it's too much to cover now, but in a so we are, we have our contact information there, but if you are strictly saying it is an aseptic process, it is not providing log reduction, then you would refer to that ISO document as very good guidance for that approach. Okay, the question here, are there methods to sterilize tissue, or can it only be aseptically processed? That is a great question, and at this point still engenders much debate. There are some countries who actually require sterilization with radiation for all tissue, and they claim that it works just fine. Now, these are largely third world countries where the level of complexity of why or how the tissue is being used is not quite the same as what we have here in the U.S. So even though I won't say across the board all tissue can be sterilized with radiation, but I think many tissues can be sterilized with radiation and still function just fine. Bone being among them. In many cases, a lot of bone is being, in different forms is being irradiated, and it seems to work very well. The critical components there are not irradiating under normal conditions, but irradiating under temperature-controlled conditions, and perhaps even oxygen control. So there are ways to reduce the amount of damage that occurs to tissue by the radiation process. And again, things that we can't go into in great detail, but we're happy to discuss on a one-to-one basis if there are additional questions there. Okay, we might have time for one more. Is there another one? Okay, how do you account for tissue variation? I'm talking a lot here. Alpha, do you want to?
No.
Okay. All right, Alpha says I should take this one as well. How do you account for tissue variation? That is a great question, and that's something that we've discussed within Nelson Laboratories to quite a bit of extent. At this point, I always refer back to first what the requirements are. At this point, there is no requirement that a particular number of different donors or different tissue types be addressed. Then that means that it is up to the tissue bank to determine what they feel is appropriate regarding different tissue types. We do not see requirements, for example, to evaluate every piece of soft tissue, every different type of soft tissue you might see or that you might process. But it is wise, certainly, in your evaluation to make sure that at least some of the main tissue types, for example, in a soft tissue scenario, that they are represented in your validation. Now, this is one situation where process characterization can be very helpful. You can use different tissue types in process characterization of individual steps where you might not be testing quite the same quantity or where smaller pieces of tissue may be appropriate. And you might find out in characterization as you're doing these different steps that it doesn't matter, or that one particular type of soft tissue is more difficult to kill because the microorganisms perhaps are somehow shielded by the tissue, or the tissue provides some type of safety for those microorganisms. And so in process characterization, it can help you understand which types of tissue to evaluate for the full validation. But at this point, generally speaking, just make sure that your evaluation covers a wide range. But we have not seen any requirement that every type of soft tissue, for example, be evaluated in the process validation. Okay. All right, and one more quick question here. Can LAL be used as a demonstration of asepsis? And that's a great question as well. So LAL is one of the test methods that we use to demonstrate presence or absence of endotoxin. The endotoxin tends to be on the surface of gram-negative microorganisms, and endotoxin will give you an endotoxin reaction whether the microorganism is dead or alive. And so evaluation of endotoxin, although important in tissue, it's not discussed very much, but it certainly is wise to make sure that you don't have large amount of endotoxin on tissue. Using it as an indicator of the amount of kill that a process has achieved would not work, because you would be seeing the endotoxin results of both the living and the dead microorganisms. Okay, Mike, turn the phone over to you.
All right, again, we'd like to thank everyone for their participation today, and especially to Martel and Alpha for presenting to us today. As you can see, Martel and Alpha's contact information there, they'd be more than happy to answer any of those questions that we did not get to today. You can contact them via email. Also, you can contact our tissue specialist, Aaron Sheeving, and he would be more than happy to assist you as much as he can as well. Again, we'd like to thank everyone for attending, and we hope that this was beneficial for you, and we hope that you have a great day. Again, you can always follow Nelson Labs on Facebook and Twitter and get updates on future webinars and even locations where Alpha and Martel may be presenting on future tissue topics. Again, we thank you and have a great day.